he will also cover some additional topics that we can address through thesis in the lab. Okay. All right. So let me start sharing my screen. I will, oh no, sorry, it's not open yet. Okay, you should see my screen now, right?
Yes, we can.
Perfect, let me just, okay. All right, so as you see from the title, most of the thesis proposal we have are on analysis of human-generated content. To me, this means that we cover any kind of content generated by people, mostly on the web platform. And I would say that clearly the main target of this analysis is social network content. But we can expand beyond that also on other sources of information. And then some of other research and thesis activities are more on to quantitative data, and I will present them separately. Okay. Just a general premise, whatever I'm going to show here is just for intuition and let's say imaginary perception of what the thesis could be in terms of broad topics. Then when we come to defining the actual topic of the work, we get in touch one-to-one and we talk about specific challenges, specific let's say objectives of each thesis work. Okay. Also consider that these are general intuitive topics, and then we can expand also beyond that, and we can intermix your let's say interests with our research objectives as well. Okay. All right. So in terms of broad contents and let's say challenges we are addressing, I would start from the first one, the first problem, that is what I would put into the broad framework of knowledge extraction. Knowledge extraction from human-generated content means this. When we look into social media or whatever other content people produce, we explore the content, but we also want to build conceptual information out of this content. So we receive a stream of text, essentially, or of any other kind of semi-structured data. Our objective is to transform this text into structured information. Imagine this as a knowledge base, imagine it as a database, whatever you like. Our objective is that of creating comprehensive understandings of the domain people talk about by looking at the text, detecting the concepts people talk about, and this is what the typical name entity extraction approaches do. But then what we are mostly interested in is to create first, detect entities that people talk about. So people, organizations, locations, time, whatever, entities that are mentioned in the discussions. Understand their frequency, their importance, their relevance. Put this entity in connection one to the other by understanding not only the entities, but also how they connect to each other, right? And also, finally, understand the typing of these entities. So understand for each entity what is its category in general, okay? This is the general framework, and the idea is, okay, we use techniques that extract and find the entities, determine the relevance of these candidate entities, And then based on that, we decide whether to keep these entities in our analysis framework or not, assign the types, and then also associate the relations. Okay, relations means that we want to actually understand that if someone talks about something in social media, it's actually talking about things or people. Okay, so in this case we have Angelina Jolie or a silk top. Okay, but then we also need to understand what's the relation between these things. And for instance, by detecting verbal phrases, we could extract the predicates that connect things, and based on that, extract the fact, a conceptual representation of that, the fact that it's a subject does something over an object. Okay, and this is new knowledge that we acquire. Okay, the idea is that through this kind of acquisition, we could detect new facts and new happenings, and then by filtering out only the relevant aspects and only the aspects that are mentioned frequently enough or that are popular enough, we could build a knowledge base of actual, say, collected new knowledge from social media. Okay, so this would be the topic of knowledge extraction from social media. The second topic I would like to talk about is instead more on the quantitative part. This is not related instead to content shared on the web from a consumer perspective, but is instead about a very special kind of content that people share, that is software artifacts. The idea is that of studying the behavior of software developers, for instance, in online open source repositories of software, study their behavior, study how the projects evolve in time, understand what's the strategies in which the projects evolve, understand what happens in the project, understand what are the typical, for instance, productivity measures and quality measures in the project, understand what are the activities that developers perform, for instance, how many people actually contribute versus how many people just watch open source, the evolution of open source projects. As you see that here in these studies, there is a pretty clear jump between the two categories. And also understand what are the network effects within and across projects, to understand what kind of communities of developers are born or are dying, are evolving in time, as well as understand the dynamics within the communities. For instance, in a previous work was focused on understanding what were, if within a software development project, we could detect some kind of peculiar management structures. For instance, the so-called rich club behavior, that is the fact that even though the project is open source, there might be a very limited club of people that are actually the ones keeping the power in their hands. And so these are the kind of researches we do. Why are they within this? Why am I presenting them here? Well, because software itself is a human-generated content, and the activities done within software communities sometimes are not so far from activities done on social media. Okay, and as you see, you actually create networks also in these sectors. All right. Then from here we expand our interests. Let's say, let me say, in the topic by moving from just analyzing the data in these kind of communities and behaviors, also to thinking about how to use machine learning and artificial intelligence to improve the software development process. So how to use, for instance, language analyzers, also implemented with deep language models for understanding and learning how people write code and improve their experience in code writing. Okay, in the same way today we experience improvement in writing tools for natural language. Okay, so the idea here is how could we move the recent approaches that have been built in language understanding for humans based on deep neural networks and so on, also to improving the activities and performance of software development. Okay. All right. Moving on. Yet another field in which is actually a very broad one, in which, in some sense, in some way, also the work presented by Francesco later may fall in, is what we could call computational social science. Here, the topic is how instead could we apply computational techniques, machine learning, artificial intelligence, big data based approaches for exploring and studying social science problems spanning from political to social to economic dynamics in society. Okay, so here I have just a couple of examples to show you, but then I can mention others, or I can point you to others. For instance, exploring and studying how elections evolve in the world in some specific settings, typically addressing the most challenging pieces. So while it's easy to predict election results in probably 80 or 90% of the cases, the tricky part is in those small parts of the countries or the society that could, by changing slightly the percentages, change completely the results of the elections. Okay, so again, here we explore social media, human-generated content in general, news, and so on. We relate to geographical aspects, and we try to understand if we can detect correlations or other kind of relations with the exogenous variables from the social ecosystem, and we understand if we see some parallelism between the two. In general, here the general framework we work on is we have the web and digital space where people express idea, opinions, and wishes, tasks, or whatever. And then we have the physical world. And the idea is, can we understand one from the other? One analysis we would like to run on this specific field is how do specific facts somehow emerge or happen as a result or as a very strong correlated effect with respect to what happens on social media. So think about recent riots in Washington before just after the elections in the month interleaving between one president and the other, and these kind of things. Here the idea is we can explore some sources of information online and see how things evolve and how things emerged along the content published and shared there. Okay. For instance, on this, we have been working, on US elections, on the Brexit phenomenon, studying, for instance, the evolution of opinions in the people before and after the, for instance, the polls, and how individual people change their mind from pro-Remain to pro-Leave and vice versa, how we could detect sentiment, stance, and other aspects, how can we intersect that with bot analysis, so whether we could detect that there are some strong biases in the presence of automated content sharing on one side or another of the political debate. Similar studies have been applied also to totally different fields, and like, for instance, studying the discrimination problems in different fields like sports, for instance, studying if there is an evidence of discrimination in different sports and across different variables, for instance, race or sex of the players, origins of the players, monetary, let's say, payment to the players, and so on. Okay. Other studies have been run over the so-called opioid pandemics pandemic in the US, the fact that there is a very strong evolution of the use of opioids and anti-pain medicine, say, treatments in the US that quickly become dependencies, and how this has been discussed and studied in social media and so on. Okay, so we have a few other topics that could be addressed along this dimension. And so more in general, okay, think of this as the general issue of relations between the digital and the physical world and the impact on society. Okay. All right. Finally, I would classify a last bunch of proposals in the field I would call of digital humanities, that instead is not so much about only studying content with respect to social impacts. It's more about studying content again, but not necessarily content in the nature of social media and social network content, but broader content in the broader sense. For instance, content coming from the so-called gamification and crowdsourcing ecosystem. This content span the use, for instance, of so-called serious games, games that are built for collecting input from users and analyzing this input for understanding some dynamics. Okay, for instance, collecting photos, collecting data, collecting opinions through games for understanding the public sentiment about a given topic, for understanding and creating together content. Okay. This field is about designing concepts of games that could be helpful for some specific objective, and then executing experiments and collecting data from these experiments and analyzing the data. As an example, I would like to mention one that we are currently running in 2 European level projects, very big ones with many top partners at the European level, for addressing problems related to policy making in general, and in particular, policy making and data analysis for the reaction to the COVID-19 pandemics. Okay, so the idea is we would build a tool that would the tool and the method, clearly, that would essentially be able to cover a very strong engagement from people, okay, by letting them not only participate by reacting to things or by, I don't know, answering to polls or questionnaires, but instead co-design possible futures, co-design possible policies, possible ideas in a participatory manner, okay, not only involving just a few people or addressing very limited and local problems, but also expanding to a large audience of people at the global level. So how could we build together ideas for solving massive scale problems, okay? Like how to solve pollution problems or plastic recycling problems, or well, also pandemics containment issues, and so on, okay? So the idea is we build tools for perceiving the feeling of the people, bringing them together and closer to a problem, and letting them contribute in a co-design approach to solve the problem, okay? This means, for instance, using online tools, platforms that let people play with images, combine content, and so on. Finally, okay, this was maybe too details, we can skip that. Again, sticking to the field of digital humanities, instead, I would skip to a completely different objective, that is, study literature. For instance, could we use NLP and machine learning, a recent advancement, to understand better how literature works actually evolve? So for instance, we pick one novel, we pick many novels, we pick any kind of fictional content, okay? And we try to understand what happens in those texts by reconstructing in a data-driven way the story written in the text. So are we able to imagine, are we able to take, for instance, a novel and build automatically a timeline of the presence of the people, how people interact with each other, what happens, what kind of events occur, in which sequence, and possibly then not study only one single book, but maybe a series of books. Okay, you take all the books of, okay, here we have an example for science fiction, Isaac Asimov, or you take all the books of Harry Potter, whatever, and you analyze how the different actors evolve in time. And clearly the final objective could be almost unreachable. That is, okay, can I understand how a role evolves its personality in time? That would be a very huge result, how to be extracted automatically, but instead much more factual aspects, like who are the people that are met more frequently, or who are the people that are contrasting each other, or when this happens, or what. This is a very interesting point. If we push it forward, it would be already great to detect things like reordering. You know, for instance, in a book you have settings like flashbacks or alternation of scenes in different timelines. Think about, I don't know, a thriller. Sometimes you have interleaved, maybe one scene about, I don't know, the crime and the criminals, and one scene about the detectives, right? and they interleave in the story, but then you could build the two parallel timelines of these things. And so the idea is this kind of work. There is already a preliminary small thesis done that, for instance, explored the Harry Potter novels and was able to build the basic connections between the actors in the story, the characters in the story. But there's really a lot to do over this kind of work. Okay. All right. With this, I think I went already quite long, so I'm done with my general proposals. In this, consider that again, along these directions, there are plenty of alternatives and options. So here I only gave you some general ideas. I wanted to go for the challenges and the problems. I didn't mention the techniques and the technical aspects. Clearly, for address these challenges, every time you need to enter specific challenges and aspects on the technical side as well. Okay, and these are discussed specifically on each separate, say, thesis at a personal level. All right. Okay, so I'm done from my part. I'm not sure if you plan to have questions now or at the end or what.
Thank you, Professor. You are right on time, and the presentation was really interesting. We usually wait a few seconds now to see if anyone has any questions. And if no one says anything, we go on with the next presentations.
Okay, I see a question about prerequisites. Well, there's no strict prerequisite. The expectation is that you have some clearly knowledge about programming and basic knowledge of data analysis, let's say, possibly some knowledge about NLP and natural language processing. All in all, consider that every thesis has an implementation experience attached. So most of the work is on actually implementing either analysis pipelines or tools or anyway some kind of development. Okay, so for sure there's coding involved, and if I'd say some basic machine learning is also appreciated because most of the analyses are also run using those techniques. Consider that basically always you have an initial phase where you have a learning curve to pass before entering the actual core of the development, but that's part of the game. Okay. Okay, I see that we also have here, by the way, Francesco is already, I guess, ready for talking about this part. I see also Stefano in the call. We can't hear you. And
so just a quick, let's say hello to everyone. I was invited to this meeting, which I normally attend. And this time I prefer that the presentation is given to Francesco, who has a lot of ideas about what to do with fake news or disinformation. And he is a PhD student of our group. So let me just introduce Francesco. That's my role today. Easy. Easy way out.
Thank you, Stefano, and thanks, Marco. And thanks everyone for the invitation. I'll just share my screen now. All right. So can you see anything?
yes.
All right. So hello everyone. I'm Francesco Pierri, and I'm a third-year PhD candidate student in an interdisciplinary PhD program called Data Analytics and Decision Sciences. And my supervisors are Professor Stefano Ceri and Professor Fabio Pammolli. And I'm currently also a visiting researcher at the Observatory of Social Media at the Indiana University, led by Professor 𝗙𝗪𝗍𝗄. And my visit is actually remote for the moment. And my research interests lie in understanding information and disinformation diffusion, social networks, and of course, complex systems in general. And I have a background in computer engineering. So in the last few years, I've been working on on disinformation spreading on online social networks mostly. So I've been investigating the presence of Italian disinformation on Twitter before the European Parliament elections of a few years ago. And I've been working also on a network-based approach, so a hybrid methodology including network science and machine learning to detect online disinformation spreading on Twitter by only looking at the way this information is shared compared to more traditional and mainstream news. And I've been also working throughout last year on understanding the COVID-19 infodemic on both Facebook and Twitter. So infodemic is a new term to indicate the disinformation and misinformation and fake news and whatever kind of low credibility information related to the COVID-19 pandemic. And on a different project, I've been working on understanding the impact of lockdown restrictions in Italy during the pandemic, of course, and with a mixture of network science and economics. And right now I'm working mostly on understanding the interplay between online conversations about vaccines and the vaccine rollout, both in Italy and in the United States. So we have produced a dashboard that you can visit using this link, and where we show several statistics about how low and high credibility information is shared in Italian conversation around vaccines on both Twitter and Facebook. So the bigger goal is to understand whether, for instance, disinformation affect the vaccine uptake in different Italian regions or provinces. And but we plan also to investigate several other signals, such as the presence of bots and coordinated inauthentic behavior on Twitter, sentiment analysis, Google search, and so on. So whatever can help to understand whether things happening on the cyber world can affect the real world. And as I said, I'm working on two different projects. One about Italy, in which Marco and Stefano are supervising, and then another at the Indiana University, where we do more or less the same thing. So we try to understand whether the vaccine campaign at the state level is affected by the presence of online disinformation. And we found, for instance, that the Republican states tend to have a higher level of misinformation, and also tend to be more hesitant towards the vaccines. So this is more or less what I'm working on right now. And I'll just go over with a list of proposals, and then whoever is interested can write to me, and we can have a heads up on this, and I can expand a little bit more. So first of all, the first thesis the first project that I'm proposing you is, of course, to understand what's going on social networks around vaccines. So the idea is to monitor the Italian conversations, but not only Italian, I mean, about vaccines, in particular the COVID-19 vaccines, with a specific focus on the spread of low and high credibility information. So I'm using here low and high credibility information to refer to, respectively, disinformation, misinformation, fake news on one side, and on the other side, more traditional and mainstream news. And some questions could be, for example, see whether we observe polarization between users, or if there are bots or other coordinated efforts to, drive specific narratives. And as I said before, at the end, understand whether there is a correlation between online content and the real-world vaccination campaign. More in general, I would like to investigate how online disinformation spreads on different platforms. So from Twitter and Facebook, which are the most, let's say, the most popular ones in the research community, but also Instagram, Parler, which is a far right-wing social network, which has become very popular after the Capitol Hill storming events, and of course YouTube. And it would be nice to understand how the same story is propagated across multiple platforms, and to detect the presence of potential super spreaders. So this is a term which has gained popularity in the context of the pandemic. You know, whenever a single individual is able to spread the virus or a specific content to a large number of other individuals, and of course to understand if there is coordination at play on different platforms. You know, a story is shared on a specific platform, and then there are individuals who take it from that platform and they move it to other platforms as well. And then another interesting topic would be to understand, so as you may know, Donald Trump has been banned from both Twitter and Facebook after the events of Capitol Hill. And he was well known as the biggest super spreader of disinformation about COVID-19 in 2020. And I believe that this would impact the spread of disinformation in general on the two platforms. So it would be interesting to understand how the agenda of disinformation website has been affected by Trump's ban, and whether there are visible effects, you know, in the media landscape. And finally, this is something a bit different. So whenever scientific papers are published or put as preprints, they're usually highly advertised on Twitter. And it might be that papers which get more visibility on Twitter, either because of a better marketing campaign, or because the authors are more popular, they might get more citations. So it could be interesting to understand whether there is a correlation between the visibility on Twitter and the number of citations that these papers get over time. And of course, it might be that there are some coordinated efforts, either authentic or inauthentic, that could include social bots. And that would be it. So thank you. And if you have any questions, please feel free to ask them.
Thank you, Professor. That was really interesting.
Thank you.
And,
I'm not a professor.
Oh, well, almost professor.
Yeah, it will be a nice long way.
Well, sooner or later, I'm sure.
I must say it's a long way, but Francesco is on the right track. That's good enough.
Thank you.
Well, now I see that Professor Massaroli is connected. So welcome, Professor. If you're ready, you can start your presentation.
Okay, welcome to everybody. I start sharing the slides. Okay, can I do it? Share content. Okay. Okay, so first of all, good afternoon to everybody. I connected just a few minutes ago, so I'm not sure what you introduced before. But anyway, I am Marco Massaroli. I am part of the big team of Professor Stefano Ceri, driven by the data-driven genomic computing project, big ERC project founded by the European Commission 5 years. And within this group, I am more focused on mostly aspect of bioinformatics and cancer genomic, as well as on aspect related to e-health. Today, I am here with Silvia. Silvia was a former thesis student of me. She graduated 2 years ago, more or less, and then she stayed within me and the group, focusing on the follow-up of her thesis, which actually is also related to the thesis proposal of today, in particular to the aspect related to patient stratification and cancer subtype identifications. So I will start making the introduction of the topic, and then I let Silvia to proceed in illustrating the proposals that are more related to the topic she is working on, since she now started the PhD with our group 3 months ago. So, as I mentioned, the proposal regards the application of data science in cancer genomics and e-health, and in particular within the framework of bioinformatics. This is nothing that should be worried about by those of you that are not aware about bioinformatics or biomolecular aspects, so did not follow my course or didn't touch this topic before, since there is a very close relationship between bioinformatics and data science. As data science, bioinformatics is the intersection between 3 major aspects, statistics, computer science, and molecular biology, or biology in general, whereas data science is more general, and the third aspect is the general knowledge of the domain to which data science is applied. So actually, in my cases, data science is applied mainly on biological and biomolecular data. Bioinformatics actually is much more than data science in respect that is not focused only on the data analysis, but also regards data management, data integration, data retrieval, and sharing, and many other aspects, which anyway I would not talk about today. But the aspect related to the evaluation of the biomolecular data are very close to the approaches that are typically used in data science. And in fact, also in the evaluation, bioinformatic evaluation of the biomolecular data, we follow the same path. So first of all, understanding the problem, the domain aspects, the biomolecular aspects, then extracting, retrieving the data typically from a remote public repository, and then evaluating the quality of those data, try to extract the sub-data which have a better quality through a classical data cleaning process. And then, after data exploration, identify the features that that characterize those data according to the problem that we want to solve. And when these features are not evident within the data, we build them by pre-processing of the data with the classical step of feature engineering, which I am sure you are very aware about. And then start the classical building of the model of the data. Typically, they are predictive models that are then applied for actually the actual evaluation of biomedical data in general, followed by their typical interpretation of the results. So the paths are very overlapping between the two, and this is the reason why I propose a thesis actually in data science with application in bioinformatics and e-health. So actually there are some background that is needed to develop this thesis, but the background is quite easy, so if you don't have yet and you are interested in the topic, can be easily acquired. For what regards the thesis applied on the cancer genomic, the background is actually related to the knowledge of the genome, of our genome, as mainly identified by the DNA, which is a sequence of many elements, actually only four different elements, but a chain of these four different elements very long. And within this long sequence, there are subsequences that are the genes that are very important because, in particular, the protein coding genes encode the information for producing within each cell of our organism first the RNA and then the sequence of amino acids that characterize the proteins. Proteins that provide the phenotypic and functional characteristics of each cell so that they provide the functionality that enable our organism to work as it does typically. The gene in our genome are about 25,000, so not so many, but then there are many other aspects that complicate the scenario. So first of all, despite all the cells in a single individual have the same, at least originally the same genome, not all the genes in all the cells are functioning at the same time, but some part of them function depending on the activity that the single cell or tissue, subset of cells, need to perform, also according to the needs of the organism and to the environment in which the organism is living. Then each of these genes actually does not encode a single protein or a single RNA, but through what we would call in computer science information compression, the encoding of a gene is such as that from a single gene, the nature derives typically more RNAs and so more different proteins, which may differ very slightly from one another, but they provide functionality that are somehow different, and this increases the possibility of the organism to interact with their environment and to be able to solve all the issues that are related with the life of a complex organism as our. In addition, the sequence of the genome and so of the genes is not always the same, but some alteration, mutations can affect the sequence in the way that the meaning of the sequence itself, so the function of the protein that are encoded, can be altered. And this is typically occurred in the case of the genomic diseases such as cancer, in which alteration of the sequence affect the activity of the gene and then so the development of the pathologies. But the idea here is that once we have understood these very few relevant aspects, then is evident to all of you that we then can take the activity of the genes that we can measure, as well as the sequence of the gene and of the genome that we can measure as well, as features of our problem of identify different types of patients. Not really to differentiate normal from pathological state patients, because this is somehow quite easy, but to identify within patients affected by the same disease, different subtype of patients that have different genomic and so biomolecular characteristics that enable them to be more effectively treated through some specific treatment instead of others, and so that they have different also prognosis and survival to the disease. On the other side, in particular for the proposal of the thesis related to 𝗙𝗫𝗏, the background is somehow more easy, is related to the knowledge of the typical activity that regards the diagnosis of the patients, the encounter between the patients and the physicians, the collection of data from the patients through typical hospitalization, examination, and other activity, data that are typically collected and stored in structured form within electronic medical record, or also in a textual form in the cases of be able then to extract this information through some also easy text mining approaches. And then also maybe considering the fact that beside this very relevant clinical and healthcare data, there are several other data related to the patient life and environment that can also be considered to better diagnose and manage the patients in its treatment path. So in this scenario are the two topics that I propose today as a thesis in data science. So the data science for cancer genomic with a couple of proposals, and data science for health. In particular, starting from this latter one, this regards the applications of reinforcement learning and either Markov model algorithm on the evaluation of the chronic kidney disease progressions based on clinical data that can be extracted from an electronic medical record of an hospital. So the focus is on chronic kidney disease, which is a disease that is very unfortunately present within our populations, and is a disease for which there is no an effective treatment that enables to the patients to recover from this disease, but is only possible to slow the progression of the disease. And the disease actually progresses from a state to the following one with a worse functionality of the kidney, up to a point when the kidney is not functioning effectively anymore, and the only possibility for the patients is that to go through a dialysis process, so to use an external kidney to purify the blood from the product of the metabolism of the organism itself. The grading of this progression at present used by the clinician is given by these systems that is actually characterize the different stages, but is not able to predict when actually there will be the needs for the patients to enter into the dialysis. So within 6 months, 1 year, 2 years, or whatever, or what's. So the idea is to apply to this type of data that can be collected typical approaches of data science that deals with the progression of time and the arrival of new data in times. So typically approaches of reinforcement learning and either Markov models. We already studied before these problems with the previous thesis of another student of computer science that made a very good work in this sense, and he developed a set of different approaches, either for classification and regressions, and at the end identify a type of extremely randomized tree as the one that provides better classifications of those patients, in particular in the prediction of the entering into the dialysis in the next 6 months or 1 year, with a slightly lower precision and accuracy in the case of more and more specific time frames. All this has been developed in collaboration with the 𝗕𝗎𝗓𝗍𝗓 Hospital, which is one of the 7 more informatized hospital in Italy, despite being a public hospital with a very well-developed electronic medical record till the first year of 2000, and from where we extracted the data that we used for these previous evaluations, which has been the main work for a paper that has been submitted and is now under evaluations. So the idea would be to use the same data that are anonymized to apply on those same data the different approaches that I mentioned, reinforcement learning and 𝗕𝗎𝗓𝗍𝗓 model, and compare the results with those previously obtained with different more classical algorithm, and try to improve the prediction also at longer time frame. Everything will be done in collaboration with the 𝗕𝗎𝗓𝗍𝗓 Hospital, but since the data are already available, and since also the previous students that worked on the development of those model is anyway available for information for any of you that are interested on this thesis and for the starting steps, the work can be done also remotely without going to the 𝗕𝗎𝗓𝗍𝗓 Hospital. In case you are interested, instead of see the activity, the informatics and data science activity within the 𝗕𝗎𝗓𝗍𝗓 Hospital, there is also this possibility to go there to work with the people, the team, the computer science team that that is in charge of this aspect in the 𝗕𝗎𝗓𝗍𝗓 Hospital, and possibly update the data that we were used with a new one, since these data that I'm referring are about 2 or 3 years old at the moment. But the path of the thesis is quite well defined. It's possible to start right away, since we also have the data available, and is also quite interesting given the fact that we apply this advanced algorithm for solving this topic. The other proposals are within the subtopic of data science applied to cancer genomics. The title are listed here, but I leave Silvia the word for their description.
Thank you, Marco. Thank you for introducing me and I'm Silvia Cascianelli. Good afternoon to all of you. And I will present you this couple of thesis proposal in data science for cancer genomics area. As Marco already told you, oh, sorry, I don't know why, but I cannot. Okay, now it's working. There's something. There's a delay. Okay.
Yes.
Sorry. Let's see if I can go back to the beginning. Anyway, as Marco told you, almost 3 years ago, I graduated with a thesis on this area, and so I had the opportunity of applying data science and machine learning approaches working on oncogenomics data. So I found it really fascinating, and also because these are two fields that I think are really, really interesting in my point of view. And in these years, we gained a lot of experience in such subjects. So these two thesis proposals are strictly related both to the studies that we have already performed and to those that we are planning to do in the next future. Particularly the first one is focused on breast cancer subtyping. That was also the topic of my thesis, but is still not a topic in the medical domain, connected with prognostically relevant patient stratification. While the second one is related to a new project that will be focused on analyzing mutational data of cancer patients, with the aim of finding actionable genes, that are those genes that could be potential therapeutic targets, and predisposing genes, that are those genes that are related to the genetic risk of having cancer. So that should be screened carefully, mainly in those cases of people with a family history of cancer. And for what concerns this latter proposal, we will focus mainly on mutational data, but with possible extension also to other relevant type of genomic data, on which we already gained experience from previous works. And we consider particularly the colorectal cancer, that is also a kind of cancer that we are studying with an ongoing thesis at the moment for what concern subtyping, because it's another cancer like breast cancer characterized by an high level of heterogeneity in terms also of expected outcomes and treatment options. And it's very interesting because many mechanisms of the onset and progression of colorectal cancer are still unknown, and there's also a great influence of genetic risk and so family history of cancer for an inherited mutation, in this case, for such kind of cancer. So, for what concern the topic of breast cancer subtyping, as said, we've worked a lot in the last few years on this topic with the aim of finding prognostically relevant patient stratification. So, starting from genomic data with the aim of, and particularly gene expression data, in such a way to help physicians in and support their therapeutic decisions, basically. Okay, it's very difficult to be sure that it works. Basically, our contribution was developing computational models to assign a patient, a single patient with an intrinsic subtype and a recurrence risk score, considering that for each patient we have available a gene expression profile, which is basically, as Marco told you, a sample, an instance made of as many features, potentially as many features as the genes under investigation. So that, of course, need a careful step of data feature selection as to find the most important and feature useful in the task of subtyping, or in the task of predicting the expected clinical outcome, that is the expected prognosis for the patients, or the recurrence event, that is the event of having again the cancer after surgery. Okay.
Mm-hmm.
Can you see the screen?
Yes.
Okay. Okay. I can. Okay. Now I can see everything perfectly. So, working on this topic in the last few years, we have produced several relevant publications on the subject. Particularly, one of them is based on the work that mainly on the work that I performed during my thesis. don't know why, Marco, but maybe it's better that you change the slide because so
if you like, you need to give me back the control.
Ah, okay. No, I think you can do it anyway, but okay, I can give you back the control.
Okay.
Sorry, I don't know why it's not working properly, but it's
okay.
Really okay. So, as I was saying, we produced several publications on the subject, starting from the publication that was related to the work, part of the work that I did during my thesis, and then following thesis always on the topic of breast cancer subtyping, as well as another relevant thesis in the focus on predicting breast cancer recurrence or relapse, that are synonyms, basically. So you can go on, please. Okay. So here, for instance, you can see that for what concerns the publication that is strictly related to the work of my thesis, we applied data science into different paths simultaneously, basically in parallel. From one side, an emulation path that as objective the trying to reproduce and trying to improve the already existing classification approaches developed for breast cancer subtyping that showed many limitations. Basically, many of them, some of them provide only dataset level classification. That basically means that you cannot provide a single patient, so a single sample with a subtype independently from a group under exam. While other approaches have proprietary algorithms and should be in principle run on proprietary platforms. So it was interesting trying to reproduce these approaches and provide scripts able to emulate their results and even improve them. On the other side, we proposed some alternative classifiers, and in a machine learning survey, basically, we compare different classifiers up to find the best option at the moment for obtaining the subtype for a breast cancer patient. But even most importantly, we focus on feature selection because, as I mentioned, we worked with quite restrained sample sizes because there's not so many patients profiled with all the useful genomic data available. But at the same time, we have a huge dimensionality of possible features. So all the possible genes, as Marco told you, around 20,000-30,000 genes for patients at the beginning. So of course, feature selection study becomes a crucial aspect in such kind of works. And this is something that this topic, okay, you can go on, was the main focus also of the following thesis, on which we were interested on trying deep approaches on such kind of task. So we compared our logistic regression baseline with feed-forward neural network, variational autoencoders, and conditional variational autoencoders, even in semi-supervised like setting, in the sense that to supply to the few availability of data of breast cancer data annotated with subtype labels, we use also extra data in our autoencoder based models, basically to try to improve and the latent space representation using data coming from other cancer type or data coming from normal breast cancer tissue. So data that are in some way related to the original one, to the actual one, that are the breast cancer ones, but useful to provide the sort of data augmentation together with these with feeding our models with extra data. We also tried approaches of data augmentation, but unfortunately, in the end, we noticed that at the state of the art, let's say, a simpler model performed better than a more complex one and deeper one, probably also because of the still restrained availability of data that can be used to feed our models. But at the current rate of increasing data availability, maybe in the next feature, it will be possible to find some improvements with these deeper models. Anyway, another important aspect that we study on this topic, working with deep approaches, is dealing with a careful step of data of result interpretation and feature interpretation. That is trying to understand the role of the features involved in the model. That is something that it's always a little bit, let's say, difficult, hard to define for deep models, and this is the reason why this kind of model are not so used in medical domain, because even more than for other domains, it's crucial that you can provide a biological and a reasonable interpretation for the predictive features, so for the feature that have been used in the model itself. So we gained some experience also on this topic. And finally, we worked also on a lot on feature selection. Again, in this case, in the for what concerned the prediction of recurrence events for breast cancer patients, we focus more on the feature selection task again. Basically developing a tripartite hybrid approach, starting from a very simple filter method, just to do a first filtering of the huge availability of feature, so of genes at the beginning. But then the core aspect of this study was to develop a wrapper method, okay, using a multi-objective genetic algorithms that wants to minimize simultaneously the prediction errors, but also the size of the selected feature space, let's say, that in our case was a feature space made of genes of interest for the classification, for the prediction task. And in the end, we also add an important occurrence-based module, okay, that takes care of aggregate results coming from the multi-objective genetic algorithms, because this kind of algorithms is a valuable resource, but it's one of its drawbacks, let's say, is that provides a set of optimal or suboptimal solutions in Pareto fronts. So it's very important to produce also module, an additional module, an additional layer of feature selection, able to condensate all the gained information and provide the relevant signatures that are basically subset of genes of interest used as feature space for our recurrence event predictors. So you can go on. So basically, what I want to let you know is that we've worked a lot on applying data science and machine learning techniques on this kind of cancer genomic topics, mostly for what concerns breast cancer subtyping or clinical prediction, but as I already mentioned, with ongoing thesis also on other kind of cancers. And all these topics are strictly related and are meaningful examples of what you can go on, is required to do in a thesis of in such area, so data science for cancer genomics. Particularly, here we have, currently we have two proposals. One is again focused on the topic of breast cancer subtyping, that, as I told you, is still a not topic in medical domain, but from a new perspective. So a perspective that to the best of our knowledge, is still quite unknown. That is the role of transcript isoforms that are basically the possible transcripts that originate from a single genes that Marco mentioned before. So their role in distinguishing breast cancer subtypes, and particularly not, we don't want simply to adhere to existing classification system, but possibly explore new data-driven classification systems, as well as other perspective of interpreting already existing standards, for instance, with a multi-label classification perspective instead of the more commonly used single label perspective. Again, always from our experience of ambiguity in some cancer samples that could exhibit mixed traits among different subtypes. On the other side, we propose also a thesis focused on a new project that is investigating mutational profiles of cancer patients to find the genes, and particularly genes variants, so genes with mutation that could be relevant therapeutic targets, or even relevantly related with genetic risk of cancer. As said, for what concerns this thesis, at the beginning, for sure, we will focus on colorectal cancer for the already mentioned characteristics. And we could also decide to conjugate the analysis and investigation of mutational profiles with a multi-omic perspective that is taking into account also other relevant kind of genomic data, as the transcriptional, so again the gene expression data, or copy number alteration. So basically trying to provide a more comprehensive view of such patients, with the aim of finding all those genes that could be associated, that could be useful both for screening patients, okay, in terms of genetic risk, or to find more suitable treatment options, moving steps towards personalized medicine, basically. So for what concerns further details about these two theses, I don't want to be too long, so you can find everything in the links that we provided in the slides. And here you have, as Marco already showed you, here you have also our contact. So for any further doubts or for asking for something, for a thesis or for something related. And of course, if you have, feel free to ask questions if you have questions.
Okay.
All right. Thank you, Professor and Silvia, for your interesting presentation. I would also like to thank everyone who took part in this event. And we are done for today, and I hope to see you in our next events.
Can I say also one thing? So I didn't
Yes,
present.
of course.
I didn't present anything because at the moment I don't have data that could be used for serious data analysis, but it may happen soon. So I think that in this case, I would use you or Francesco in order to propose some potential thesis, but I prefer not to do it now because in the lack of data, it is difficult to do anything. So it is reasonable not to propose a thesis without data. But as I said, it could happen quite soon.
All right, you can send me an email and then we'll discuss it.
Yes, okay, good.
Shall we send to you the PDF of the presentation?
Yes, thank you.
Okay, I'll do. Thank you to you.
You're welcome. All right, so bye everyone.
Bye-bye.
Bye.